welcome to the deep dive. Today we're tackling something really foundational based on these pretty detailed academic notes you've brought in. We're looking at the cell.
That's right, the absolute bedrock of biology. You really can't understand life without getting into the nitty-gritty of the cell.
And our plan isn't just to, list definitions. We want to piece together the bigger picture from these notes, how cells are organized, how they specialize.
And crucially, how they manage everything that comes in and goes out. Traffic control, basically.
Exactly. We're aiming for a real understanding of cell theory, the organelles inside, and those vital transport mechanisms, going beyond the flashcards.
Well said. Because understanding the cell, it's really about understanding complexity itself. We'll focus on what makes different cells tick and the elegant structures doing the work.
Okay, so foundation first. Cell theory. Where do we start?
Right, the three big rules. You really have to internalize these. First,
all living things are made of cells. Got it.
Yep. Second, the cell itself is the basic unit of life, the smallest thing that is life.
Makes sense.
And third, kind of mind-blowing when you first hear it, all cells come from other pre-existing cells. They don't just pop into existence.
Defines life right there. Okay, so with those rules in place, the notes then immediately jump into a big division, right? Prokaryotes versus eukaryotes.
Yes, a fundamental split. And it's not just about size, it's about internal organization, or, well, lack thereof in one case.
Prokaryotes, the name means before the nucleus, right? So simple structure.
Very simple. No true nucleus for their DNA. Always single-celled organisms. And a key thing, no complex internal skeleton, no cytoskeleton.
Okay, so then eukaryotes. True nucleuses. These are the complex ones.
These are the ones most people picture. They have that membrane-bound nucleus protecting the DNA. They often build multicellular creatures like us.
And they do have that internal scaffolding, the cytoskeleton.
Yeah.
That seems like a huge leap.
It was a huge leap. It likely enabled all the complex life we see. And how that happened, well, that leads to a really fascinating idea.
Ah, you mean the endosymbiosis theory. I remember reading about this.
Exactly. The idea is that some of the key components inside our eukaryotic cells weren't originally part of the cell. They were separate, free-living prokaryotes themselves.
So like a big cell swallowed a smaller one.
Kind of. Imagine a larger prokaryote engulfing, say, an ancient bacterium that was really good at generating energy, like a precursor to mitochondria.
Okay.
But instead of digesting it, it just stayed, took up residence, became a permanent partner.
And the same for chloroplasts in plants.
That's the theory. They were originally photosynthetic bacteria. It's incredible because it explains why mitochondria and chloroplasts still have their own DNA and their own ribosomes.
Right. They can sort of replicate themselves inside the main cell.
Precisely. They carry the echoes of their independent past. It's evolution happening at this tiny internal scale.
That idea of specialization is huge. And it continues when we look just within eukaryotes, comparing, say, plant cells and animal cells.
Absolutely. They share a lot of the basics, nucleus, mitochondria, ER, but they have distinct features tailored to their lifestyles.
Let's take plants first. The notes highlight that super rigid cell wall.
Yeah, made of cellulose. It's like structural armor, gives them shape, protection, prevents them from bursting if they take on too much water.
And they manage storage and energy differently too, right?
Yeah.
Plastids.
Right. Chloroplasts are the famous ones for photosynthesis, but plants also use others, like leucoplasts to store starch, or chromoplasts for color in flowers and fruits. Different jobs, different plastids.
And the giant water balloon inside, the central vacuole.
The central vacuole, yeah. It dominates the plant cell interior, stores water, nutrients, waste, and it's key for maintaining turgor pressure, keeping the plant cell firm.
Okay, so plants, rigidity, storage, energy capture. What defines animal cells in contrast?
Animal cells are more about dynamism, movement, and cleanup.
Like centrioles for cell division.
Exactly. Centrioles, part of the centrosome, organize the spindle fibers. These are like ropes that pull the chromosomes apart accurately when the cell divides. Plants manage this differently, using organizing regions, but animals have these specialized structures.
And the cleanup crew, lysosomes.
Lysosomes, yes. Think of them as the cell's demolition and recycling centers. They're filled with these powerful hydrolytic enzymes, basically digestive juices.
So they break down food particles or old, worn-out organelle.
Exactly. Intracellular digestion. But they also have a more, well, dramatic role.
Apoptosis. Programmed cell death.
That's the one. If a cell is damaged, infected, or just old, the lysosomes can be triggered to rupture inside the cell.
Whoa.
Yeah, releasing those enzymes to digest the cell from within. It's a controlled self-destruction, very important for development and preventing disease.
Okay, let's zoom in on two powerhouse organelles mentioned heavily in the notes. First, the mitochondrion, the energy factory.
The absolute powerhouse, site of cellular respiration where glucose gets converted into ATP, the cell's energy currency.
And its structure is key. You mentioned the endosymbiosis link again.
Definitely. It has that double membrane, a relic of being engulfed, but the inner membrane is where the action is. It's highly folded.
Into cristae.
Cristae, yes. These folds massively increase the surface area. And embedded all over that surface are the enzyme complexes needed for making ATP. More surface area, more energy production. It's incredibly efficient.
So the mitochondrion makes the power, but then there's the whole manufacturing and shipping department, the ER and Golgi.
Right, the endoplasmic reticulum, or ER. It's this vast network of interconnected membranes, like tunnels and sacs throughout the cytoplasm.
It comes in two types, rough and smooth.
Correct. Rough ER looks rough because it's studded with ribosomes.
Ribosomes make proteins, so rough ER is all about protein synthesis and modification.
Primarily, yes. Proteins destined for secretion or insertion into membranes are made there. Then you have the smooth ER. No ribosomes, hence smooth.
And it does other stuff. The notes list quite a bit.
It's a chemical wizard. Smooth ER synthesizes lipids like steroids. It metabolizes carbohydrates. And critically, it handles detoxification.
Detox, like breaking down toxins.
Exactly. Things like alcohol, drugs, metabolic byproducts. Liver cells, which are detox powerhouses for the body, are packed with smooth ER for this reason.
Okay, so ER makes proteins and lipids and detoxifies. Then things move to the Golgi.
The Golgi apparatus, or Golgi complex. Think of it as the cell's post office and finishing center. It receives proteins and lipids from the ER.
What does it do with them?
It modifies them further, sorts them, tags them for their final destination, and packages them into vesicles, little membrane bubbles.
Like shipping containers.
Perfect analogy. If a protein needs to be secreted out of the cell, the Golgi packages it into a secretory vesicle, which then travels to the plasma membrane and fuses with it, releasing the contents outside.
Amazing coordination.
Yeah.
But none of this intricate movement or even holding shape would work without internal support, right? The cytoskeleton.
Absolutely critical. The cytoskeleton isn't just passive scaffolding, though. That's key.
You said it's dynamic.
Very dynamic. It's constantly being assembled and disassembled. Yes, it provides shape and mechanical support, but it does so much more.
Like positioning organelles.
Yes, anchoring them or moving them around. And it creates tracks, like highways, for motor proteins to walk along, carrying vesicles and other cargo.
So it's the cell's internal transport network, too. The notes break it down into 3 fiber types: microfilaments, immediate filaments, and microtubules.
Right. Let's focus on the two most involved in movement and structure. Microfilaments are the thinnest. They're made of actin.
Actin, like in muscles.
Exactly the same protein. They work with myosin for muscle contraction, but they also help cells change shape, crawl like an amoeba or a white blood cell, and they form the contractile ring that pinches a cell in two during division. Very versatile.
Okay, thin and involved in pinching and crawling. What about microtubules?
Microtubules are the largest ones. Hollow tubes made of a protein called tubulin. They are the main structural components and the highways for intracellular transport.
Big, strong tracks.
Yes. And they also form the core of cilia and flagella, those whip-like tails or hair-like projections that help some cells move.
Like sperm cells.
Sperm tails are classic flagella, built from microtubules. They also form the spindle fibers during cell division, the ones organized by the centrosomes we mentioned earlier.
Okay, so we've got the internal structure and highways sorted. Now, the boundary itself, the plasma membrane.
The keepers.
Yeah.
It's not just a passive bag, it's an active interface. Its defining feature is selective permeability.
Meaning it chooses what gets in and out?
Precisely. It controls passage based on the cell's needs, not just, say, the size or charge of the molecule or whether there's more inside or outside. It's highly regulated.
And the structure that allows this is the fluid mosaic model. Sounds fancy.
It describes it perfectly. It's fluid because the components can move around laterally, it's not rigid, and it's a mosaic because it's made of different pieces.
The main piece being the phospholipid bilayer.
That's the foundation. 2 layers of phospholipid molecules. They have heads that like water hydrophilic, pointing outwards and inwards towards the cytoplasm, and tails that hate water hydrophobic, tucked away in the middle.
Creating a fatty barrier.
A barrier that water-soluble things can't easily cross. But embedded within this lipid sea are large protein molecules.
The channels and pumps.
Exactly. They provide pathways for specific substances. And scattered throughout, you also find cholesterol molecules.
What do?
They regulate fluidity. Cholesterol acts like a buffer, prevents the membrane from becoming too fluid at high temperatures or too solid at low temperatures. Keeps it just right.
Okay, so that's the structure. How does the actual transport work across this selective barrier? The notes start with passive transport.
Right. Passive means no energy required from the cell. Molecules move simply because of differences in concentration.
Down the gradient, from high concentration to low concentration.
Always. The simplest form is simple diffusion. Small, nonpolar molecules, like oxygen or carbon dioxide, can just slip right through the lipid bilayer, like oxygen moving from your lungs into your blood.
Easy enough. What if something needs help?
That's facilitated diffusion. The molecule still moves down its concentration gradient high to low, no energy needed, but it needs a protein channel or carrier to help it across the membrane, like ions or glucose sometimes.
Still passive, just needs a doorway.
Correct. And a really crucial type of passive transport is osmosis.
The movement of water.
Specifically, the diffusion of water across a selectively permeable membrane. Water also moves from where it's more concentrated, meaning fewer solids, to where it's less concentrated, more solids.
So if you put a cell in really salty water, that's a hypertonic solution.
Yes. Hypertonic means higher solute concentration outside. Water will rush out of the cell towards the higher salt concentration.
And the cell shrinks, plasmolysis.
Exactly. It shrivels up. The opposite happens in a hypotonic solution, lower solute concentration outside. Water rushes in.
Which could make an animal cell burst.
It certainly can, since they lack that rigid cell wall plants have. Osmosis has major consequences for cells.
Okay, so that's passive. But cells sometimes need to move things the other way, right?
Yeah.
Against the gradient.
Absolutely. Sometimes a cell needs to accumulate something that's already more concentrated inside or pump something out that's less concentrated outside, moving uphill against the concentration gradient.
And that takes energy.
That requires energy, usually in the form of ATP. This is active transport.
How does it work for big stuff? The notes mention endocytosis and exocytosis.
Right. These are for bulk transport. Exocytosis is how the cell exports large molecules or particles. Remember those vesicles from the Golgi?
Yeah.
They move to the plasma membrane, fuse with it, and spill their contents outside the cell. That's exocytosis. Secretion.
And endocytosis is the reverse, bringing stuff in.
Exactly. The cell membrane folds inwards, engulfing material from the outside, and pinches off to form a vesicle inside the cytoplasm.
The notes list 3 types. Phagocytosis.
Phagocytosis is cell-eating, engulfing large solid particles like a white blood cell engulfing a bacterium.
Penocytosis.
Cell drinking, taking in droplets of extracellular fluid and whatever solutes are dissolved in it, less specific.
And receptor-mediated endocytosis.
This is highly specific. The cell membrane has receptor proteins that bind to particular molecules outside, like cholesterol bound to carrier proteins. Once enough receptors are occupied, the membrane invaginates, bringing in a concentrated dose of that specific substance. Very efficient.
So vesicles for bulk. What about smaller things like ions against the gradient?
That often relies on specific protein pumps embedded in the membrane, ion pumps.
Like the sodium-potassium pump. The notes really emphasize this one for nerve cells.
It's fundamentally important. This pump uses ATP energy to actively push sodium ions out of the nerve cell and potassium ions in. Both movements are against their respective concentration gradients.
Constantly working, constantly burning ATP.
Constantly. Maintaining this imbalance is crucial for nerve function, for generating electrical signals. It's a prime example of active transport at work.
Wow. Okay, so pulling this all together. We started with the basic rules, cell theory. We saw the split between simple prokaryotes and complex eukaryotes, maybe thanks to endosymbiosis.
Right, incorporating those ancient partners.
Then the specialization plant walls and vacuoles versus animal lysosomes and centrioles, the internal powerhouses like mitochondria, the Golgi factory, and post office.
All supported by that dynamic cytoskeleton highway system.
And finally, the selective membrane controlling all the traffic, using both passive flow and energy-demanding active transport like those pumps.
You've got it.
It really drives home that the cell isn't just some bag of chemicals. It's this incredibly complex, dynamic, self-regulating system. A fortress, like you said earlier.
It really is. And think about that last point, active transport. We saw how concentration gradients drive passive movement naturally.
High to low, yeah.
But cells spend a huge amount of energy, like with the sodium-potassium pump, specifically to maintain an imbalance, to keep sodium high outside and potassium high inside the nerve cell.
Right, fighting against diffusion.
Exactly. So the final thought to leave you with is why. Why is actively maintaining that specific chemical imbalance, that stored potential energy across the nerve cell membranes, so absolutely critical for everything we do, for thinking, moving, feeling?
It's the foundation for nerve impulses, isn't it? That stored difference is what allows the signal to fire.
Precisely. The energy spent creating that imbalance is the potential that powers our entire nervous system. It highlights how life isn't just about reaching equilibrium, it's often about actively maintaining disequilibrium to get work done.
A fantastic point to ponder. That's all the time we have for this deep dive. Thanks for joining us.
My pleasure. Fascinating stuff.